On the recording, the change is almost imperceptible at first. The trace that had been moving in loose, rounded waves, slow oscillations hovering around 8 to 12 cycles per second, the rhythm of a mind still loosely tethered to the room, begins to slow. The waves spread apart. They flatten slightly at their peaks. Then, in a shift that takes only a few seconds to complete, the rhythm drops below 8 cycles per second and keeps descending, the peaks growing longer and gentler, the whole trace settling into the slower, wider undulation of theta. In the darkened lab, on a monitor no one is urgently watching, a brain has just crossed the threshold from waking into sleep. It is not a dramatic event. There is no single moment of crossing, no clean edge between one state and the other. The transition is gradual and electrical, a slow change in the dominant frequency of millions of neurons settling from one rhythm into another, the way a crowd slowly quiets, not all at once, but in a gradual softening until the room has become something different from what it was a minute before. The researcher watching the trace has seen this hundreds of times. The change is ordinary and exact. Alpha fades, theta arrives, the brain has begun. What happens next, across the hours between that first moment of descent and the slow brightening of morning, is not rest in any simple sense. It is a sequence of distinct electrical states, each with its own architecture, each with its own signature visible on the trace, cycling through in a pattern that repeats with quiet regularity all night long. The sleeping brain is not still, it is organized, and the organization, once seen, has the quality of something that has been doing this for a very long time. The first stage passes quickly. Theta rhythms persist for a few minutes, the brain in a light, drifting state, easy to pull back, the sleeper still close to the surface. Then something changes again. The trace shifts, and in that shift, one of the most distinctive signatures in all of sleep recording appears. A spindle is a small thing, measured in seconds. On the trace, it looks exactly as the name suggests, a symmetrical burst of oscillation that begins almost imperceptibly, builds toward a brief spinning peak, and then tapers off again into the baseline rhythm. The whole event lasting less than 2 seconds from first ripple to last. The frequency is fast for sleep, between 11 and 16 cycles per second, a busy little tremor nested inside the slower theta that surrounds it. It appears, completes itself, and is gone. What generates a spindle is a loop. Deep inside the brain, tucked within the structure of the thalamus, there are neurons whose task during this stage of sleep is rhythmic inhibition. They fire, and because of how they are connected, their firing silences the cells they are talking to, and then that silence releases them, and they fire again. The loop sustains itself. The oscillation propagates outward through thalamocortical connections, reaching the surface of the cortex and appearing on the EAG trace as that brief waxing-waning burst. Then it fades. The loop settles. The trace returns to its baseline. Spindles come in clusters. They punctuate the slow rhythm of stage 2 sleep like a repeated motif in a long piece of music, arriving, completing, dissolving, arriving again. Mixed among them, at irregular intervals, are a different kind of event altogether. A K-complex is large. On the trace, it stands out immediately, a sharp negative wave, followed by a slower positive deflection, the whole structure taller and broader than anything surrounding it. A single biphasic event that can be triggered by a soft sound from the environment, or that can arise from within the sleeping brain on its own. Its appearance is abrupt. Its purpose is not entirely settled. The current understanding is that it may suppress the cortical activity that would otherwise wake the sleeper, a dampening signal issued by the brain to itself, holding sleep together against small intrusions. Between spindles and K complexes, the trace continues its slow roll. The sleeper is in the shallows still, but the brain has organized itself into something new. This is not waking. It is not the deepest sleep. It is stage 2, a structure all its own, recognizable to anyone trained to read the record, occupying the largest fraction of a full night's sleep by total time. Then, in the first cycle of the night, usually within the first hour, the trace changes again. The spindles thin out, the K complexes become less frequent, and in their place, spreading slowly across the trace, come waves of a different character altogether, slower, wider, higher in amplitude, rolling across the EEG record like low swells across open water. These are delta waves. They are the signature of the deepest sleep the brain produces. Their frequency is low, between half a cycle per second and 4 cycles per second, and when they occupy more than a fifth of a scored epoch, the record has entered slow-wave sleep. Inside the brain during slow-wave sleep, the activity is extraordinary in its scale. Neurons across wide territories of the cortex are synchronizing their behavior in a way that has no equivalent in waking. There is a moment, repeated, cyclic, occurring about once per second, when a vast sweep of cortical neurons shift into a state of high activity, firing together at near-waking rates, a wave of collective excitation that produces the tall positive slope of the delta wave on the trace. Then, almost simultaneously, those same neurons hyperpolarize. They go quiet. Not just a few of them, millions of them, across broad regions, pulling their membrane potentials below the threshold for firing and holding there. The trace dips, the wave turns. A brief collective silence falls across the cortex, and then the cycle begins again. Slow-wave sleep is front-loaded into the night. The deepest and longest bouts of delta activity come early, concentrated into the first 2 cycles, the first 3 or 4 hours of sleep. The delta waves that appear in those early cycles are tall and broad, their amplitude at its highest, their dominance within each scored epoch at its greatest. If a researcher were to plot only the slow-wave sleep content across the full night, they would see a shape that descends steeply into the first cycle, rises somewhat for the second, and then progressively shallows across each subsequent cycle, the waves growing shorter and narrower with each repetition, the deep, slow sleep gradually giving way to something lighter, something that becomes, by the last cycles of the night, barely a trace at all. This front-loading is not arbitrary. It is a reflection of something that has been building since morning. Throughout every waking hour, a molecule called adenosine accumulates in the spaces between neurons, produced as a byproduct of the brain's energy expenditure, diffusing slowly into the synaptic environment, accumulating molecule by molecule in a way that cannot be hurried or reversed by any act of will. By late evening, after a full day of waking, adenosine levels in the brain's interstitial spaces have reached a threshold at which their pressure on the sleep-regulating circuits becomes irresistible. That pressure is the feeling of heaviness in the eyes, the slowing of thought, the loosening of attention. It is process S, the homeostatic drive built from the accumulated cost of being awake. The two-process model, described in 1982 by the researcher Alexander Borbeli, gives this homeostatic drive its formal frame. Process S rises during waking and falls during sleep. It is countered and shaped by Process C, the circadian clock, the internal 24-hour rhythm that determines when the body is prepared to sleep and when it is prepared to be alert, independently of how tired the organism is. The interaction of these two processes, the homeostatic pressure and the circadian timing, determines when sleep arrives, how deep it runs, and how it distributes itself across the night. They are not the same thing. A person can be exhausted by accumulated adenosine and still lie awake if the circadian clock is signaling the wrong time, and a person can feel wide awake in the early afternoon precisely because the circadian alerting signal is strong enough to temporarily suppress the homeostatic pressure that has been building since morning. Slow-wave sleep discharges, process S. As delta waves move through the sleeping brain, adenosine is cleared from the synaptic spaces. The pressure dissolves, slowly, across the first cycles of the night. By the time the deepest slow-wave sleep has passed, by the time the third cycle begins and the delta waves are already shorter and less frequent, a large portion of the homeostatic debt has been paid. The brain's need for the deepest sleep has for the time being been met. And what comes in its place, filling the later cycles of the night in progressively longer episodes, is something entirely different. The first REM episode of the night is brief. It arrives at the end of the first cycle, roughly an hour and a half after sleep onset, and it lasts only a few minutes before the cycle turns and the brain descends again towards slow-wave sleep. But the trace during those few minutes looks nothing like what came before. On the EEG, REM sleep produces a pattern that resembles waking more closely than it resembles the slow waves of stage 2 or the broad delta of slow-wave sleep. The waves are low in amplitude, the frequency is high. The signal is irregular, varied, close to the desynchronized trace of an alert and active brain. A researcher encountering this pattern without any other information might initially believe the sleeper had woken, but two other things in the record tell a different story. The first is the eye movement channel. During REM, the electrodes placed near the eyes record bursts of rapid conjugate movement, the eyes moving quickly behind closed lids, tracking nothing in no consistent direction, driven not by any external stimulus, but by the activity of the brain's own circuitry. The movements are brief and clustered, separated by periods of quiet. They give the state its name. The second is the EMG, the muscle activity recording. During waking and during most of NREM sleep, the muscles of the body maintain some residual tone. During REM sleep, they lose it, not partially, almost completely. The skeletal muscles, the ones that move limbs and trunk and all the voluntary apparatus of the body, go slack. This is not simply relaxation, it is active paralysis imposed by a circuit in the brainstem. In the pons, a dense compact region of brainstem tissue sitting just above the medulla, specific nuclei issue a chemical command that travels down the spinal cord and suppresses the motor neurons that would otherwise activate the muscles. The body of the sleeper lies still, while the brain above lights up into something close to its waking state. The two systems, the one that generates the brain's activity and the one that would act on it, have been decoupled at the level of the spinal cord. The irregular breathing in the EMG and respiration channels during REM confirms what the EEG already suggested. The cardiorespiratory system, which keeps to a steady, regular pattern during slow-wave sleep, becomes variable and unpredictable during REM. Heart rate shifts. Breathing deepens and quickens and slows. The whole metabolic profile of the sleeping body fluctuates in a way that tracks some internal, unseen activity rather than the stable, slow rhythms of deep sleep. That first REM episode ends, the cycle completes, and then the second cycle begins, the brain sliding back down from REM towards slower rhythms, but not descending as deeply as before. The slow-wave sleep in the second cycle is shorter than in the first. the delta waves less dominant, the bout briefer. The homeostatic pressure that drove the deep, slow wave sleep of the first cycle has already been partially discharged. Process S has declined. Process C is ticking through its cycle, and as the night continues, the balance between the two processes shifts in a way that reshapes the architecture of every subsequent cycle. By the third cycle, slow wave sleep has become much reduced. By the fourth, it may be absent almost entirely. The brain, as the second half of the night arrives, has shifted its center of gravity away from the deep delta states and toward something else, toward REM, which in each successive cycle grows longer, fuller, and more elaborate. The second REM episode lasts longer than the first, the third longer still, and by the final cycle of the night, in the last hour or two before natural waking, the brain's activity during REM has expanded into something that can last nearly an hour, a sustained period of low amplitude, high-frequency EEG activity, rapid eye movements, complete muscle, atonia, irregular breath and heartbeat, the whole metabolic signature of a brain deeply engaged with its own inner processes, while the body beneath it lies perfectly still. The 90-minute cycle is a rough average, and real cycles vary. The first cycle of the night often runs a little shorter. Later cycles stretch slightly longer as REM episodes extend, but the rhythm holds with enough regularity across individuals and across nights that it can be traced clearly in overnight recordings. The cycling of the brain from light to deep to REM and back again, repeating 4 or 5 times across 8 hours, like a slow, regular breathing of the neural architecture itself. There is an image that helps hold the shape of a single cycle in the mind. Think of the brain as a structure that over 90 minutes traces a gradual descent and return, dropping from the light theta rhythms of early sleep down through the spindle-dotted terrain of stage 2, then further into the broad, slow landscape of delta, and then turning slowly back upward through lighter NREM stages, and finally into the brief, vivid, near-waking activity of REM before cycling again. The descent is unhurried, the ascent is gradual, and each completed cycle brings the brain back to a starting point that is itself slightly different from where it began. The homeostatic pressure a little lower, the circadian clock a little further through its arc, the next REM episode a little longer than the one before. From one angle, a night of sleep looks like 5 repetitions of the same 90-minute pattern. From another, it looks like a single slow arc, the night opening with the heaviest, deepest slow wave sleep the brain will produce in those hours, and then, across successive cycles, the center of gravity shifting, the delta growing shorter and shallower, the REM growing longer and more elaborate, until the final cycle arrives and the whole balance has reversed, and the brain spends the last hour of the night not in the deep quiet of slow wave sleep, but in the high activity of its longest REM episode. The architecture is not an accident of biology. Each component of the cycle has its own generators, its own oscillatory mechanisms, its own distinct relationships to the rest of the brain's activity. Sleep spindles emerge from thalamocortical loops. Delta waves reflect the synchronized up and down cycling of broad cortical territories. REM is maintained by a circuit in the brainstem whose inhibitory signals suppress waking, whose cholinergic activity sustains the desynchronized EEG, and whose descending commands paralyze the body's muscles with quiet precision. The shift from waking to sleep, from alpha to theta, is itself a measurable event, a change in the dominant oscillatory frequency of the brain's electrical activity, crossing a threshold that EEG scoring systems define with specific criteria. The alpha rhythm of relaxed waking, roughly 8 to 12 cycles per second, gives way to the slower, broader theta waves of stage 1, below 8 cycles per second, and this transition is reliable enough and consistent enough across individuals that it has become one of the primary markers used to identify sleep onset in research and clinical recordings. Below the theta of stage 1, the spindle-rich architecture of stage 2 begins. Spindles arrive within the first few minutes and recur throughout the stage at irregular intervals, each one generated anew by the thalamocortical loop, completing its self-sustaining oscillation, each one fading as the loop dissipates. A full night of stage 2 sleep produces hundreds of individual spindles, hundreds of brief, symmetrical bursts of 11 to 16 cycle oscillation, waxing and waning on the trace, each one lasting less than 2 seconds, each one a ripple spreading and dissolving across the electrical surface of the sleeping brain. The K-complex punctuates these spindles less frequently, but with greater amplitude. It arrives suddenly, a sharp, tall wave that dominates the trace for a second or two before fading back, and then the spindles resume, and the long, slow baseline rhythm of stage 2 continues. There is something almost protective in the rhythm of it. Spindles arrive, K-complexes hold sleep together against small disturbances. The brain moves through the stage in a quiet alternation of oscillatory events, none of them lasting more than a few seconds, all of them expressing the underlying thalamocortical architecture that distinguishes this stage from all the others. Then the delta waves arrive, and everything grows quieter. Not in terms of electrical activity, the delta waves of slow wave sleep are the highest amplitude waves on the EEG trace, larger than anything in stage 2, their broad peaks visible even to untrained eyes as the dominant feature of the record. But the quality changes, the spindles thin out, and the K-complexes disappear. The trace settles into the slow, rolling regularity of the deep, slow oscillation, the brain moving in long, broad waves that are measured not in cycles per second, but in fractions of a second per cycle. The whole rhythm so slow that the gap between one peak and the next is nearly 2 seconds. Inside that rhythm, the upstates and downstates of the cortex are cycling in slow alternation. The upstate brings a broad wave of collective neuronal firing, a moment of high activity across millions of cells. The downstate that follows is the opposite, a hyperpolarized silence, brief and nearly complete, a pause in the collective activity of the cortex, so thorough that on the EEG trace it appears as a clear negative deflection, marking the valley between one wave and the next. And then the upstate returns, and the cycle continues, one complete oscillation per second or slower, maintained across the full duration of slow wave sleep. The deepest slow wave sleep of the night is in the first cycle. The delta waves that appear in that first descent, usually within the first 45 minutes or hour of sleep, are at their tallest, their broadest, their most dominant. A researcher scoring an overnight record can see immediately which part of the night they are in by the amplitude of the slow waves. The first cycle's slow wave sleep has a characteristic weight and breadth to it, a depth of signal that distinguishes it from the shallower delta of later cycles. As the night progresses and adenosine clears and the homeostatic pressure of process S declines, the slow waves of subsequent cycles grow progressively lower, less frequent, less dominant within each epoch. By the fourth or fifth cycle, slow wave sleep may contribute only briefly, a handful of delta-rich epochs, before the brain rises again toward lighter sleep and then into REM. The progressive shortening of slow-wave sleep across successive cycles and the progressive lengthening of REM are two sides of the same shift. As process S declines and its homeostatic pressure for deep slow-wave sleep diminishes, the balance tips toward the conditions that favor REM, conditions partly determined by the circadian clock, which in the early morning hours is running at a phase that supports longer and more stable REM episodes. The interaction of these two processes produces the characteristic shape of the night. Slow-wave sleep, heavy at the beginning, REM light and brief, then, across successive cycles, slow-wave sleep retreating and REM expanding, until in the final cycle, the night has become almost entirely REM episode. The pons is small. It sits at the base of the brain, where the forebrain transitions into the narrowing structure of the brainstem, a compact, dense region of tissue that does several essential things simultaneously, and whose contribution to REM sleep is fundamental. During waking and NREM sleep, the descending motor pathways are intact. Signals from the motor cortex reach the spinal cord and the muscles respond. At the transition into REM, something changes in the pons, specific nuclei within it, glycinergic and GABAergic neurons, whose axons run down the lateral tegmentospinal tract, begin to fire, and their firing suppresses the motor neurons of the spinal cord below the level of voluntary control. The muscles of the limbs, the trunk, the face relax and go slack in a paralysis that is active, maintained, and reversed only when REM ends. What this means in the context of the sleeping body is that during each REM episode, the most metabolically active state the sleeping brain enters, the state in which the EEG resembles waking and the eyes move rapidly, and the heart and breathing rates fluctuate. The body attached to that brain is as close to motionless as it ever is outside of anesthesia or deep surgical paralysis. The brain ignites into near-waking activity, and the body lies still. The two systems have been separated at the level of the brainstem by a circuit operating automatically in the dark, and the result is a sleeper who is physiologically still while neurologically vivid. The REM episodes of the last cycles of the night are the longest, and they are the most physiologically complete. The early REM episodes, the ones that appear briefly at the end of the first and second cycles, are shorter and sometimes contain periods of quiet eye movement activity and lower metabolic engagement. The final REM episode, the one that fills the last hour before natural waking, has a different quality. It is sustained. The eye movements come in longer bursts. The cardiorespiratory variability is more pronounced. The EEG desynchronization is thorough and maintained. The paralysis holds, and the brain, during this long final REM episode, is running close to its waking metabolic rate, drawing on glucose and oxygen at near-waking levels, doing something internally that requires the same energy expenditure as being awake. What that activity is, what the brain is doing in those long final REM episodes, is a question that sits at the edge of this picture. The electrical record shows what the brain is doing. It does not show why. The EEG trace tells a researcher that the brain is running in a high-frequency, low-amplitude, desynchronized state with rapid eye movements and muscle atonia and cardiorespiratory irregularity. It tells them when this state occurs and how long it lasts and how it changes across the night. The deeper questions, what is being processed, what is being preserved, what the metabolic activity of that long final REM episode is for, sit just beyond what the trace alone can answer. The two-process model gives a framework for the timing. Slow-wave sleep is built by the homeostatic pressure of adenosine and discharged by the delta activity of early sleep. REM is shaped by the circadian clock's promotion of the conditions that support it, concentrated into the late night and early morning hours when Process C is running at a phase favorable to REM, and Process S has declined enough to allow the brain to sustain it fully. The interaction of these two rhythms, one built from the accumulated cost of waking, one running on an internal clock independent of that cost, produces, across a full night, a structure that is not random and not fixed, but dynamically organized, responsive to how long the person has been awake and to what time it is, and resolved into a consistent arc, deep slow wave sleep in the first half, progressively longer REM in the second. The cycling is regular enough to be predictable. Most adults, sleeping normally in the absence of sleep debt or circadian disruption, will complete 4 or 5 cycles across a full night. Each cycle covers roughly 90 minutes. Each cycle ends in a REM episode that is longer than the one before, and each successive REM episode is more elaborate, metabolically more active, more fully developed in its electrical signature than anything that came earlier in the night. The final REM episode is the longest the brain will produce until the next night arrives. It appears in the last hour or two before natural waking, filling that portion of the night with its characteristic desynchronized activity, its rapid eye movements, its muscle atonia, its irregular breath and heart rate. The brain at this point has been cycling through NREM and REM for 6, 7, 8 hours. The homeostatic pressure of Process S has been discharged by the slow wave sleep of the early cycles. The circadian clock is running in the phase of maximum support for REM, and the sleeping brain, in its last extended episode before consciousness returns, runs its most elaborate and metabolically demanding activity of the entire night. There is something in the shape of this, the way the night begins in the heaviest, quietest deep sleep and ends in the most vivid, energetic REM, that has the quality of an ordered progression, not a climax, nothing so dramatic as that, but a natural sequence, one state giving way to another, the homeostatic and circadian processes moving through their arcs and producing, as their interaction, an architecture that has remained consistent across human sleep for as long as there have been recordings to read. The trace that began in the early minutes with a slow shift from alpha to theta, a gradual electrical quieting, an unremarkable descent into the shallows of stage 1. That trace has moved across 7 or 8 hours through spindles and K complexes, and the broad rolling delta of slow wave sleep, and the brief frequent REM of early cycles, and it has arrived at last at the long vivid near-waking activity of the final REM episode, the brain running at near full speed while the body lies still, the night nearly done, the morning close. On the recording, the trace of that final REM episode is busy and low and fast, the signature of a brain doing something elaborate and sustained, something it saves for the end of the night. The delta waves are gone, the spindles are gone. The broad, slow architecture of the night's first hours has given way entirely to this high-frequency activity, this metabolic investment, this last long state before the alpha returns and the threshold back to waking is crossed. The researcher in the darkened lab, if they are still watching, would see the trace change once more as morning arrives. The low-amplitude, high-frequency activity of REM slowly gives way, and the broader, slower waves of relaxed waking return, alpha 8 to 12 cycles per second, the brain recrossing the threshold it first crossed hours ago. The spindles are gone, the delta is gone, the REM has run its course, the night has completed its arc, and in the hour before that final waking, in the long sustained, metabolically active REM episode that occupies the last portion of the night, The sleeping brain has been doing what it does at the end of every night, running its most elaborate state, drawing on near-waking energy, doing something the trace records but does not fully explain. The night that began in the smallest electrical event, a slow shift in frequency, a trace dropping below 8 cycles per second in a darkened lab, has ended with the brain at its most active, its most vivid, saving something intricate and sustained for last, just before the light comes back. Deep in the membrane of a single neuron, in a channel so narrow that only one ion can pass at a time, a magnesium atom sits lodged in place like a stone pressed into a gap in a wall. The channel is called an NMDA receptor. It is one of hundreds of thousands distributed across the branching surface of this one cell, and across the billions of cells in the brain, the same structure appears again and again, the same narrow throat, the same blockade, the same waiting. The magnesium does not move unless two things happen at once. Glutamate must bind from outside, arriving on a signal from the cell upstream, and the membrane around the channel must become electrically charged enough, depolarized enough, to push the magnesium free. Both conditions together, neither alone is sufficient. This is the cellular meaning of learning, not a simple switch, but a double requirement, a gate that opens only when two signals meet, a lock with two keys. When the gate opens, calcium floods in. It is a brief, steep surge, calcium ions moving fast down their concentration gradient, drawn through by the electrical difference between outside and inside the channel. The surge lasts only moments, but in those moments, the calcium activates a cascade of enzymes within the cell. The enzymes begin moving AMPA receptors, a different kind of receptor, one that responds to glutamate without a double requirement, toward the surface of the synapse. More AMPA receptors at the synapse means more sensitivity. When the upstream cell fires again, the connection grows stronger. It is a change that begins in a fraction of a second, and a change that, given the right conditions, can persist for years. This is long-term potentiation. It is the leading mechanism by which the brain converts a fleeting experience into something that can be retrieved later. Its name is formal and technical, and yet what it describes is one of the most ordinary things the brain does: the quiet thickening of a synapse's response to a signal it has received before. The strengthened synapse is the brain's pencil mark, the faint physical record that something happened here, that this cell and that cell fired together, that the pairing was worth reinforcing. It is not memory yet. It is the raw material from which memory will be made. The chain of events that opens a calcium channel in a dendritic spine, and the chain of events that allows a person to remember where they were on a particular afternoon, are separated by so many steps that it can seem impossible. They are the same process. They are. The calcium surge triggers protein kinases. The kinases phosphorylate existing AMPA receptors, making them more conductive. They also activate signals that travel to the nucleus, where they trigger the production of new proteins, structural proteins that grow new synaptic connections over the hours that follow. A dendritic spine, the tiny physical protrusion that forms one half of a synapse, can be built from nothing in the hours after strong learning. It grows along the branching tree of a neuron's surface, small and new and structurally committed, a material record of something that occurred in the world outside. These spines are too small to see with the naked eye. They are a fraction of a hair's width in diameter. Under an electron microscope, they appear as small rounded swellings at the tips of thin shafts projecting from the main dendrite, each one a site where a connection has formed or strengthened. After a potent learning event, new spines appear. They are not permanent. Many will be pruned back in the hours ahead. Some will survive and become part of the lasting architecture of a memory. But in the hours immediately after learning, the dendrite's surface grows briefly more populated, more elaborate, more ready. Somewhere above this molecular activity, above the single synapse, above the single neuron, there is a structure the rough size and shape of a bent finger, curved and tucked deep in the temporal lobe on each side of the brain. It receives information from all the major sensory and association areas of the neocortex, gathering together what was seen and what was heard and where the body was in space and what emotional coloring the moment carried, all arriving along a great convergent highway called the perforant path through a gateway structure called the entorhinal cortex. The structure that receives all of this is called the hippocampus, and its job is to bind it together. The binding is not simple. The signals arrive already processed, already transformed from raw sensory input into something like abstracted representation, the cortex's version of the event. The visual cortex has already recognized the face. The auditory cortex has already parsed the sound. The prefrontal cortex has already tagged the moment with context and expectation. What arrives at the hippocampus is not the raw world, but the brain's encoding of it. And what the hippocampus must do is take these separate streams, arriving through separate pathways, representing separate aspects of the same moment, and link them into a unified, retrievable trace. When any one part of the experience is encountered again later, the hippocampus can use that part to reconstruct the whole. This is what a memory is at the circuit level, a pattern of activation that can be restarted by a fragment of itself. Inside the hippocampus, there is a region called the dentate gyrus. It is the hippocampus's first major processing stage, the first place the arriving signals are transformed. And here the brain does something that seems almost paradoxical. To form memories that can be distinguished from one another, it operates at extreme sparsity. On any given experience, only a small fraction of the dentate gyrus's granule cells, somewhere between 2 and 5 in every 100, become active. The rest stay silent. This is not a failure of encoding. It is the mechanism by which similar memories are kept distinct. Because the active cells are so few, and because which cells are active is specific to each experience, two similar experiences can be represented by almost entirely non-overlapping patterns of activity. The woman at the cafe on Tuesday and the woman at the cafe on Wednesday do not blur into a single memory. They are encoded in patterns that share almost nothing, even though the experiences shared almost everything. The dentate gyrus separates them by forcing each into its own narrow constellation of firing cells, each constellation distinguishable from every other. This process is called pattern separation, and it is the reason that dense urban memory, the layered accumulation of a life lived in one city, in one neighborhood, in one repeating daily sequence, does not collapse into an undifferentiated mass. The brain does not file experience alphabetically or chronologically or by category. It files each moment into a unique sparse neural pattern and keeps those patterns apart by using only a thin sliver of its available cells to represent any one of them. From the dentate gyrus, the signal moves deeper into the hippocampus through a region called CA3, where the architecture allows each cell to connect with a huge number of other CA3 cells, a structure built for pattern completion, for taking a degraded or partial input and finding its way to the full pattern. Then the signal moves to CA1, and from there back out, back toward the entorhinal cortex, and eventually toward the neocortex. The hippocampus is not a dead end. It is a loop, a way station, a coordinator. It receives from the cortex, processes, and sends back. At the cortex, the same experience is also being registered, but differently. The neocortex's version of the trace is distributed and weak. The visual cortex holds a faint impression of the visual content. The auditory cortex holds something of what was heard. The prefrontal areas hold the contextual frame, the sense of when and why. These impressions are thin and labile in the first hours after encoding. They cannot be retrieved on their own. They are like faint pencil marks on a large surface, scattered and difficult to find without a guide. The hippocampus, for now, is that guide. Its binding trace holds the addresses, so to speak, of the cortical impressions scattered across the broader brain. When retrieval is attempted, the hippocampus reconstructs the experience from those distributed fragments, pulling them together again into something resembling the original pattern. The retrieval is not playback, it is reconstruction. And reconstruction is only possible as long as the hippocampal trace remains intact. A memory in this state, freshly encoded, hippocampus-dependent, cortically distributed, but only weakly held, is described in the language of consolidation theory as labile. The word is borrowed from chemistry, where it describes a compound that is easily altered or broken down. A labile compound can be changed by heat, by light, by a competing reaction. A labile memory can be changed by interference, by disruption, by competing information, by the simple passage of time without reinforcement. It is held in place for now. It is not yet stable. The synaptic changes that underlie it are chemical and structural, but they have not yet been written in the deepest ink. A memory's early fragility was first observed not in a laboratory, but in people, patients who suffered damage to the hippocampus through injury, through surgery, through the long, slow attrition of disease, lost the ability to form new memories while often retaining access to older ones. The pattern was consistent: recent memories gone, remote memories preserved. The hippocampus was necessary for new learning and for the early retrieval of recent experience. But with time, the older memories seemed to leave it, to become independent of it. They had been consolidated into the neocortex itself, where they no longer required the hippocampus to be found. What this implied was that the hippocampus's role was temporary. It was a scaffold, not a foundation. It held the structure up, while the permanent support was built beneath it, and then the scaffold could come away. The model that grew from these observations is called the standard consolidation model. It proposes that memories begin in the hippocampus, dependent on it for both storage and retrieval, and gradually transfer to the neocortex over time, becoming less hippocampus dependent and more cortically distributed as they age. The process is driven by repeated reactivation of the memory trace, each reactivation strengthening the cortical connections a little more, until eventually the cortex can retrieve the memory on its own. The question the model immediately raises, and the question that has driven decades of sleep research, is what drives the reactivation? What provides the repeated signal that transfers the memory from its temporary hippocampal home into the stable cortical architecture? The neocortex is not passive in this process. It is a vast columnar interconnected structure covering the brain's outer surface. Its billions of neurons organized into layers and regions, each with specialized functions, connected by long-range fibers that allow distant regions to communicate. A memory stored here is not kept in one place. It is distributed, smeared, in the technical sense, across a constellation of regions whose collective activation recreates the experience. The visual component lives in visual cortex. The narrative component lives in regions associated with language and sequence. The emotional tag lives in areas connected to the amygdala and anterior cingulate. When a remote memory is retrieved, All of these regions activate together, coordinated by some structure that holds their connectivity, and over time, that coordinating structure shifts from the hippocampus toward the prefrontal cortex and toward the connections between the cortical areas themselves. The transfer takes time. It takes more time than waking provides. Something about the waking state, its continuous stream of new input, its demands on attention and executive function, its chemical environment limits the conditions under which consolidation can proceed. A brain that is constantly encoding new experience is a brain with limited capacity to consolidate what was just encoded. The signal-to-noise ratio is unfavorable. The very mechanisms that allow new learning, the sensitivity of synapses, the readiness of the hippocampus to form new bindings, are also the mechanisms that create interference with what was just stored. A newly formed memory trace is vulnerable to the next experience arriving over the same channels, along the same pathways, through the same neurons. The brain cannot hold still long enough to let the ink dry. There is another way to say this. The brain encoding a new experience at noon is in a different state than it needs to be to consolidate the experience from 9 in the morning. It is busy, its attention is outward. Its chemistry, norepinephrine, acetylcholine, the broad arousal of the waking modulatory systems, is oriented toward the external world, not toward the internal work of strengthening and transferring what it has already stored. Consolidation, the kind that moves a memory from the hippocampus to the cortex and writes it there in something more permanent than a fresh LTP trace, seems to need quiet. It seems to need inwardness. It seems to need a period when new input stops arriving, and the systems that were busy encoding can turn their attention to the material they have collected. This is not a metaphor. It is a hypothesis that has accumulated a great deal of evidence. The hypothesis is that the brain's consolidation mechanism is not simply time-dependent, that the relevant interval between encoding and stable storage is not waking time, but sleep time, and that the physiological conditions of sleep, its particular electrical rhythms, its chemical environment, its structural alterations in communication between brain regions, provide exactly the quiet and the architecture that consolidation requires. But that evidence and the machinery behind it belongs to a different set of hours. For now, in the fading afternoon of the waking day, the newly encoded traces sit distributed across hippocampus and neocortex, held together by connections that are strong enough to be retrieved, but not strong enough to be permanent. The dentate gyrus has filed each experience into its own sparse pattern. The CA3 and CA1 circuits have processed and relayed the signals. The cortex has registered faint distributed impressions. The NMDA channels have opened and closed. The calcium has surged and receded. The AMPA receptors have been moved to the synapse surface. The dendritic spines, some of them, have begun the slow structural work of forming. The day's experience is written. Not deeply, not yet permanently. It is there in the synaptic changes of the hippocampus, held in the electrical potential of the circuit, waiting in the molecular aftermath of the calcium surge. It is there the way a message is there when it has been typed but not yet sent, complete in its content, coherent in its form, but still requiring something to make it final. What the waking brain has produced is a draft, precise, detailed, organized by the dentate gyrus into distinct patterns, bound together by the hippocampus into unified retrievable traces tagged by the cortex with sensory and emotional context. A draft, nonetheless, that the waking brain alone cannot finalize. Consider what has been built across a single waking day. In the morning, a face, a name, a conversation, each one a separate pattern in the dentate gyrus, each one bound by the hippocampus, each one leaving its faint impression in the relevant cortical areas. By afternoon, dozens more, the street walk twice, the taste of something eaten, the sequence of steps in a task newly learned, the emotional weight of a moment when something went wrong or unexpectedly right. Each has its own constellation of active cells. Each has left its own pattern of synaptic changes. The hippocampus has been binding and linking all of them, holding the addresses, maintaining the connections. By evening, the hippocampal binding system is carrying a large load. It is not unlimited in its capacity. The dentate gyrus can separate patterns from one another, but as the day progresses and the number of stored patterns grows, the space between them narrows. Older memories risk being overwritten by newer encodings, especially when the new experience shares neural resources with the old. The interference is not catastrophic. The pattern separation of the dentate gyrus defends against it, but it is real and it accumulates. Every hour of continued waking is an hour during which the freshly encoded traces are vulnerable. There is something almost tender about the newly encoded memory trace in this state. It is complete enough to be retrieved. It is specific enough to be distinct. It is held in a cellular architecture that took millions of years of evolution to refine in a process of remarkable precision, and yet it is also fragile in a way that feels almost incongruous with the mechanism's elegance. The calcium surge that opened the NMDA channel lasted milliseconds. The AMPA receptors moved to the synapse surface within minutes. The dendritic spine, if it grows, will take hours to solidify, but the trace itself, the unified hippocampus-bound pattern that constitutes a retrievable memory, exists for now in a state of genuine precariousness, dependent on the continued integrity of a set of synaptic weights that have not yet been consolidated into the deeper, slower, more permanent architecture of the cortex. The hippocampus maintains this precarious holding state with great fidelity across the waking day. It replays traces briefly, spontaneously, even during waking pauses, during quiet rest, during mind wandering, during the gaps between active tasks. Each brief replay is a small act of reinforcement, a small accumulation of additional LTP at the relevant synapses, a small reduction in the trace's vulnerability. But these waking replays are incomplete. They occur in a noisy environment, competing with ongoing encoding. They are brief and irregular. They do not reach the deep organizational quality that the memory trace needs to transition from hippocampal to neocortical storage. The neurons in the hippocampus that held the trace of a waking experience, a place navigated, a face seen, a conversation had, carry that trace through the remaining hours of the day and into the approach of darkness. The molecular markers of the calcium surge have faded, but the structural changes they triggered are still in progress. The new AMPA receptors are in place. The dendritic spines are still forming or retracting. The hippocampal binding trace is still active, still retrievable, still guiding reconstruction when the experience is brought back to mind. But it is not finished. Something the waking brain has not yet provided, some condition, some interval, some state of altered physiology and chemistry and electrical rhythm is needed before the trace can settle into the cortex as something genuinely stable. The waking brain, for all its precision, has written only in pencil. The marks are there, specific and careful. They can be read, they can be followed. They are not yet permanent. The last light of the waking day leaves the hippocampus holding what it holds, all the patterns of the day, each one separate and distinct, each one bound and addressable, each one waiting. The cortex holds its faint, distributed impressions, thin as watermarks. The dendritic spines stand along the branching surfaces of neurons, some of them new, some of them strengthened, all of them still in the slow process of deciding whether to persist. The AMPA receptors sit at synaptic surfaces, moved there by the calcium surge of hours ago, their presence the molecular memory of a moment that is already beginning to feel distant. This is where the day's encoding rests, as evening comes and the light fades and the signals from the outside world begin to thin. The whole accumulated record of the waking hours, stored in cells too small to see, in electrical patterns too fast to feel, in molecular changes too subtle for any introspection to reach. A record of the day written in the only language the brain has, the language of synaptic weight and firing pattern and sparse cell constellation, held now in a temporary state, awaiting something the waking brain cannot provide. The requirement is not simply time. Hours of continued wakefulness do not strengthen the hippocampal trace or transfer it to the cortex. They may even weaken it as interference accumulates and the neural resources the hippocampus uses are repeatedly drawn into new encoding. What is required is a change of state, a shift in the brain's chemistry, its electrical activity, its relationship to the outside world, a period when the inward work of consolidation can proceed without competition from the ongoing demands of experience. The newly encoded memory is complete in its content. It is specific in its neural pattern. It is held with fidelity in the hippocampal binding circuit, and it is genuinely unfinished, waiting as the waking day ends for the conditions that will allow it to become permanent. It has been written, it has not been filed. That is the work still ahead, as the brain tips slowly under the accumulated pressure of a long waking day toward sleep. What lies just across the threshold of sleep is a different physiological world, a world of different chemistry, different electrical rhythms, different communication between brain regions. The hippocampal traces that are liable now will pass into that world, still held in their precarious state, still fragile, still in pencil. What will happen to them on the other side of waking? What the sleeping brain does to the draft the waking brain has made is a story written not in the language of molecules and channels, but in the larger, stranger language of oscillation and replay, of coordinated rhythms between structures that barely speak to one another while the brain is awake. For now, the day's traces rest where they are, in the hippocampus, in the faint cortical impressions, in the molecular aftermath of calcium and AMPA receptors and growing spines. The pattern separation of the dentate gyrus has done its work. The binding of the hippocampus holds. The cortex has its watermarks, and all of it, the whole accumulated archive of the day's experience, waits in the cellular quiet, incomplete, held gently in place, the way a breath is held just before sleep comes, full and still, and not yet released. Across a wide territory of cortex, millions of neurons fire together for a fraction of a second, then go quiet in unison, a wave of electrical silence rolling through the sleeping brain like a slow tide pulling back from shore. On the EEG trace, this moment is visible as a sharp negative deflection followed by a broad positive return. The cortical slow oscillation, cycling at roughly half a wave per second, too slow to seem like much of anything until the full picture comes into focus and the scale of what it is doing becomes clear. The firing is the up state, the silence is the down state, and in the transition between them, hidden inside a rhythm so gradual it almost seems like rest, a filing system of extraordinary precision is doing its work. The downstate is not a pause in activity. It is a specific, actively maintained condition, one in which neurons across a broad stretch of cortex are driven below their resting potential, held there, hyperpolarized, not permitted to fire even if a signal arrives. During waking, the cortex is rarely this quiet. Individual patches of it may quieten when attention shifts, but the wholesale coordinated suppression of firing across wide territories is something the cortex does only in slow-wave sleep, only in the dark hours of the early night, and only when the conditions are exactly right. Seen from the outside, through a set of electrodes resting gently on the scalp, this wave appears as the tallest, most rhythmically regular feature on the overnight recording, a broad, unhurried oscillation that sweeps the brain at roughly the pace of a slow breath. The upstate follows, and this is where the filing work begins. When the cortical neurons are released from hyperpolarization and allowed to fire, they do so in a brief, intense burst, not random, but patterned, shaped by whatever information the brain has been carrying since the day before. The upstate is a window of excitability. It opens, and for a fraction of a second, the cortex is poised, receptive, capable of receiving and encoding what arrives into it. Then the downstate rolls through again, quieting everything, and the cycle begins once more. Half a wave per second, 30 slow oscillations a minute. Across the long hours of the early night, hundreds of them move through the sleeping brain in sequence, each one opening and closing the window, each one part of the same patient, repetitive operation. What arrives into that window is not random either. Nested inside the slow oscillation, coordinated with its rhythm in a relationship so precise it was decades before the full picture came clear, are the two other rhythms that complete the system. One comes from the thalamus, a structure roughly at the brain's center, dense, deeply interconnected, involved in nearly every signal that passes between the body and the cortex. The other comes from the hippocampus, small, curved, buried in the medial temporal lobe, the structure that received the day's experiences and bound them into retrievable form. Together, these three rhythms, the slow oscillation, the thalamic spindle, and the hippocampal ripple, form the machinery of memory consolidation during sleep. None of the three works alone. The power comes from the way they fit together. The spindle is the middle piece. It originates in a thin shell of inhibitory neurons that wraps around the thalamus like a sleeve. A structure called the reticular nucleus, made entirely of cells that release the inhibitory signal GABA. During slow-wave sleep, these cells fall into a rhythmic pattern. They fire, inhibiting the relay neurons in the thalamus beneath them, and then briefly release that inhibition, allowing the relay neurons to rebound and fire in return. That rebound feeds back and triggers the reticular neurons again, and the cycle repeats. What emerges from this loop is a self-sustaining oscillation in the range of 11 to 16 cycles per second, fast enough that on the EEG trace it appears as a spindle-shaped burst, waxing from nothing to a peak and then falling back to silence within 1 or 2 seconds. The shape gives it its name. The symmetry of it is precise. Each spindle is a brief self-contained rhythm generated by the thalamus and broadcast upward into the cortex. On the recording, a spindle looks like a tiny storm, a compact cluster of waves building and fading against the longer, slower background of the slow oscillation, and it occurs within a very specific phase of that background. Spindles appear predominantly during the upstate, in the window when the cortex is excitable and poised to receive. This is not coincidental. The timing is a structural feature of the coupling between thalamus and cortex during slow-wave sleep. a feature that positions the spindle exactly where it can do the most good. What the spindle does, once it arrives in the cortex, is create a condition of heightened synaptic plasticity. The 11 to 16 cycles per second oscillation is fast enough to activate calcium-dependent signaling cascades in cortical neurons, the same cascades that underlie the structural changes associated with memory storage. During the brief window of each spindle, the cortex is not merely receiving signals, it is primed to retain them. The faster the spindle frequency, the more reliably this priming appears. And the spindles that appear over the rear, upper surface of the head, the centroparietal region, show the strongest correlation with memory improvement the following morning. These spindles, the fast ones, are the ones most tightly linked to what happens next. What happens next is a ripple down in the hippocampus in the curved, layered circuit that runs from the structure called CA3 into the adjacent region called CA1. A brief burst of high-frequency activity ignites. It lasts no more than 150 milliseconds, sometimes much less, sometimes only 50. But within that narrow window, it contains something that cannot quite be described by the word oscillation, because that word does not capture what the activity is carrying. The cells that fire during a hippocampal sharp wave ripple do not fire randomly. They fire in sequence. They fire in the specific order in which they were active during a waking experience earlier in the day. The finding that established this was made in 1994 in a study by Matteo Wilson and Bruce McNaughton that used tiny electrodes implanted across the hippocampus of rats as they ran through a maze. Each location in the maze had its own small cluster of cells, called place cells, that fired when the animal was in that spot. As the rat moved through the maze, the sequence of place cell activations traced its path, one location coding cell after another in the order the animal moved through space. Then the rat slept, and during sleep, in the brief bursts of hippocampal ripple activity, the same sequences fired again, the same cells in the same order, but compressed in time. A journey that had taken minutes to complete was replayed in a fraction of a second. The brain was not resting, it was reviewing. This time-compressed replay is the hippocampal contribution to the system. During the sharp wave ripple, the neural sequence that represents a waking experience, a path through space, a conversation, a face seen in a particular context, is briefly reinstated in the hippocampus. The reinstatement is not perfect, and it is not static across the night. The sequences shift slightly with each replay, losing some details, strengthening the relationships between elements that appeared together consistently. But the core of what was encoded is there, firing again in the dark, carrying the pattern of the original experience out of the hippocampus and toward the structures that will eventually hold it permanently. The direction of that transfer is upward and forward, toward the cortex, and within the cortex, toward the frontal regions, especially a region near the midline called the medial prefrontal cortex. This structure, in animals with newly formed memories, shows relatively little activity when a learned association is retrieved. The hippocampus carries most of the retrieval load in the early days, after learning, but across nights as sleep consolidation proceeds, medial prefrontal activity at retrieval gradually increases. The hippocampus becomes less involved. What was a hippocampus-dependent memory is slowly becoming a cortex-dependent one, stored not in the temporary, convergent architecture of the medial temporal lobe, but distributed across the wider neocortical system that holds knowledge over the long term. The mechanism by which the hippocampal ripple achieves this transfer is the third layer of the nested hierarchy. At the moment the ripple fires, it broadcasts its compressed sequence outward, and it does so precisely during the window when both the cortex and the thalamus are in position to receive and retain it during the up state of the slow oscillation inside the burst of a sleep spindle. The ripple is timed to arrive when the cortical gates are open, and the slow oscillation is orchestrating the timing of everything, holding the spindle in its upstate window and the ripple inside the spindle's trough, so that all three fire in coordinated sequence, nested one inside the other like rhythms of different timescale, meshing into a single coherent signal. Imagine three clocks ticking in the same room. The slowest ticks once per 2 seconds, the slow oscillation. The second ticks 14 times per second, the sleep spindle. The third fires in a burst, so rapid it completes hundreds of cycles in a single tick of the second clock, the hippocampal ripple. In an uncoordinated system, these three rhythms would have no special relationship to each other. They would overlap randomly and miss each other as often as they aligned. But in the sleeping brain, during slow-wave sleep, they do not overlap randomly. The slowest clock gates the timing of the second gates the timing of the third, and the third, firing in the moment when the gates are open on all three timescales simultaneously, delivers the memory-bearing sequence into a cortex primed to receive and retain it. The elegance of this arrangement is not incidental. Cross-frequency coupling, the precise temporal coordination of oscillations at different frequencies, is a known feature of neural communication in many contexts. But the three-way coupling found in slow-wave sleep between hippocampal ripples, thalamic spindles, and cortical slow oscillations represents a particularly clear example of the principle because the functional consequence of the coupling is so well supported. When the coordination is intact, memory consolidation proceeds. When it is disrupted by age, by certain drugs, by sleep fragmentation, the overnight improvement in memory performance is reduced. The timing is the mechanism. The slow oscillation itself had a less visible history than the spindle or the ripple. EEG recordings could detect something in the very low frequency range during slow-wave sleep for decades before its significance was clear. But the technology was not sensitive enough to resolve the fine structure of the upstate and downstate, and the oscillation was easy to confuse with slow artifact in the recording. It was in the early 1990s, using high-density EEG arrays and more precise slow drift electrodes, that the full picture of the cortical slow oscillation emerged, a rhythm generated by the cortex itself, propagating across the brain in waves that travel from front to back with each cycle, coordinating large territories of cortex into the same phase at the same time. This propagation is part of what makes the slow oscillation so well suited to its role. A signal that coordinates only a small patch of cortex would have limited reach. The slow oscillation travels. It moves through the brain with each cycle, and as it moves, it pulls the thalamus and the hippocampus into its rhythm, not simultaneously everywhere, but in a coordinated sweep, so that the spindles and ripples nested within it follow the wave as it travels, creating a moving front of consolidation that works its way across the night's sleep from one structure to the next. The evidence for the functional importance of this system came in part from experiments that interfered with it, carefully, non-invasively, using technology that could reach inside the sleeping brain without breaking its surface. Jan Born and his colleagues in Germany developed a technique called closed-loop transcranial stimulation, in which scalp electrodes monitored the brain's natural slow oscillations in real time, and a precisely timed electrical current of approximately 0.75 cycles per second was delivered through the same electrodes at exactly the moment and phase to augment the brain's own rhythm. The current was too weak to generate neural activity on its own. What it did was push the existing oscillation slightly further in the direction it was already going, a small hand on the back of a process already in motion. The effect on memory was measurable. Participants who learned a set of word pairs in the evening and then slept, with the closed-loop current amplifying their slow oscillations during non-REM sleep, recalled significantly more of the learned pairs the following morning than participants who slept without the current. The EEG confirmed that the stimulation had increased slow oscillation amplitude and, critically, had also increased spindle density within the upstates of those oscillations. The nested coupling had been strengthened by pushing only the outermost layer of the hierarchy. The memory improvement followed directly from the improved coordination. What this demonstrated was not merely that slow oscillations are associated with memory consolidation. It demonstrated that the slow oscillation was causally contributing to it. When the rhythm was amplified, when the timing was pushed further into coordination, the outcome was better retention. The consolidation system is tunable. The frequency of the slow oscillation is specific. A current delivered at 0.75 cycles per second, improved declarative memory. A current delivered at 5 cycles per second, theta frequency, not the slow oscillation's range, had no such effect. The benefit depended on matching the brain's own rhythm, augmenting the specific oscillation that carries the nesting hierarchy. In an adjacent set of experiments, the same group examined what happened when the current was delivered slightly out of phase, timed to push against the slow oscillation rather than with it. The memory effect reversed. Recall the next morning was reduced compared to unmanipulated sleep. The system's dependence on precise timing was visible even from outside the skull, through the crude medium of electrical fields applied to the scalp. The brain's filing system requires its own specific clock, and even a small disruption of that clock's phase makes the files harder to retrieve. There is something in the spatial organization of this process that rewards careful attention. The slow oscillation does not treat all memories equally. Its propagation across the brain follows a front-to-back gradient during waking learning. Regions that were most active during encoding show the highest slow-wave activity during the subsequent sleep. This spatial correspondence is not perfect, and much of the detailed mapping is still being done, but the directional principle is consistent. The brain's consolidation activity during sleep tends to be concentrated in regions that were most engaged during the day's learning. The filing system is not filing randomly. It knows, in a purely physical sense, which shelves received new material. The hippocampus during this process is not merely broadcasting, it is also listening, in the sense that the cortical upstate, when it arrives, feeds signals back into the hippocampal circuit, and this bidirectional communication, hippocampus sending compressed replays outward, cortex sending signals back during the upstate. The whole exchange, repeated hundreds of times across the night, is the substrate of what theorists call the hippocampal neocortical dialogue. The word dialogue is a metaphor, of course. There is no conversation in any meaningful sense. What there is a repeated exchange of signals in a coordinated temporal structure, with each cycle of the exchange incrementally updating the strength of specific synaptic connections in the cortex while the hippocampal trace is consulted one more time. The active systems consolidation theory, developed principally by Jan Born and his colleagues over the past 2 decades, describes this process in its full proposed form. In this account, newly formed memories enter the hippocampus during waking as rapidly encoded fragile traces, compressed representations that can be retrieved but are vulnerable to interference and decay. During slow-wave sleep, The hippocampus repeatedly activates these traces through the sharp wave ripple mechanism, sending compressed versions of the memory outward into the cortex. The cortex, primed by the spindle and coordinated by the slow oscillation, incorporates each transmission, slightly strengthening the relevant synaptic connections each time. Over the course of many cycles, across one night, and then the next, and the nights that follow, the memory is gradually transferred. The hippocampus becomes less involved in retrieval. The cortex becomes more capable of retrieving the memory on its own. What the medial prefrontal cortex looks like in this account after a week of sleep-consolidated learning is quite different from what it looked like after the original encoding. Before consolidation, retrieval of the learned information activates the hippocampus strongly and the prefrontal cortex weakly. After consolidation, the pattern is reversed. The medial prefrontal cortex responds reliably. The hippocampus is less consistently engaged. The same memory, now accessible from a different substrate, has been moved, not erased from the hippocampus, but duplicated and strengthened in the cortex until the cortical representation is robust enough to sustain retrieval without referring back to the original site of encoding. This transfer is slow because it must be slow. The cortex stores information in distributed, overlapping patterns of synaptic strength across large populations of neurons. Writing a new memory into that structure without disturbing the memories already there requires incremental change, tiny adjustments to synaptic weights that accumulate across many repetitions without ever making any single large alteration that might overwrite what is already stored. The hippocampus is designed for rapid encoding. It can bind a new memory in a single experience. The cortex is designed for stable long-term storage. It requires gradual change. The sharp wave ripple, repeated hundreds of times across the night, bridges these two systems by providing many small signals rather than one large one, a slow accumulation of inputs into a system that needs time to adjust. The same reasoning applies to why the most intense slow-wave activity is concentrated in the early part of the night. Slow-wave sleep is front-loaded. The first two sleep cycles of the night contain the deepest, most prolonged periods of delta-wave activity, with the largest and most regular slow oscillations and the highest density of sleep spindles. By the third and fourth cycle, slow-wave sleep is shorter and lighter, the oscillations less regular, the spindles less frequent. This temporal distribution is not arbitrary. At the start of the night, the hippocampal stores from the day are full. Everything that was encoded during waking is waiting to be transferred, and the pressure to consolidate is at its highest. The system runs hardest when there is most to do, and then eases across the night as the most urgent material has been transferred and the remaining rounds of the dialogue become incremental refinements. A clearest natural demonstration of what this system can do comes from the observation that humans who sleep after learning consistently retain more than those who remain awake for the same interval, an effect found across dozens of studies and multiple types of declarative material. The improvement is not a simple matter of forgetting less during sleep. The sleeping brain does not merely protect memories from interference by suspending experience, it actively improves them. When participants learn a task to a criterion level in the evening and are then tested again in the morning, they perform better on the morning test than on the final practice trial of the evening. The memory has grown stronger overnight, without any additional practice, purely as a result of what happened during sleep. The slow oscillation, the spindle, and the ripple ran their nightly program, and the trace in the cortex came out stronger on the other side. The temporal coupling of the three rhythms that makes this possible has been measured in human participants using high-density EEG confirming that the cross-frequency nesting visible in rodent recordings is present in the human brain as well. In people who show the strongest overnight memory improvement, the coupling between spindles and slow oscillations is tightest, the spindles most precisely timed to the up state of each slow oscillation cycle. In people who show less overnight improvement, the coupling is looser. The filing system's effectiveness, in other words, is not uniform across individuals or across nights. It depends on the precision of the temporal coordination, which varies with age, with sleep quality, with the architectural integrity of the slow oscillation itself. Older adults provide a particularly clear illustration of this. Normal aging brings a progressive reduction in slow wave sleep duration and slow oscillation amplitude. The tall, broad delta waves that characterize the first 2 cycles of a young adult's overnight recording become smaller and less regular across the decades, not absent, but diminished. Sleep spindle density declines as well, as does the precision of the coupling between spindles and slow oscillations. And these changes are mirrored by reduced overnight memory consolidation in healthy older adults who show smaller sleep-dependent gains on declarative learning tasks compared to younger participants, even when total sleep time is similar. The same architecture is present, but less precisely tuned, and the filing system works less efficiently as a consequence. The sharp wave ripple is also subject to disruption by the conditions of sleep, fragmented sleep, sleep interrupted by noise, by pain, by the brief arousals that characterize sleep apnea, breaks the continuity of the consolidation process at the level of the ripple. Each arousal terminates the slow wave episode in which the ripples are embedded, interrupting the nesting hierarchy and forcing the system to rebuild the conditions for transfer when sleep resumes. The number of ripples per hour of sleep is reduced. The amount of material transferred per night decreases. The memory traces that were waiting in the hippocampus at the start of the night do not move as far as they would in uninterrupted sleep. There is a quality to the slow oscillation that is worth staying with for a moment, not the mechanism, but the character of it. It is in some sense the brain's most fundamental sleeping rhythm. It arises from the intrinsic properties of cortical neurons themselves, from the way these cells cycle between an active phase and a resting phase when the neuromodulatory signals that keep them awake have been withdrawn. It does not require input from outside the cortex to sustain itself, though it is shaped and coordinated by the signals it exchanges with the thalamus and hippocampus. It is in that sense what the cortex does when left largely to itself in the dark, a slow collective oscillation between firing and silence, endlessly repeated, carrying the brain's own memories through the dark from one structure to another while the body is still. The down state, when seen in this light, is not simply an absence of activity, it is the reset, the moment of clearing, of low synaptic background, of reduced noise that makes the upstate useful. Without the down state, the upstate would have no contrast against which to write. The silence is part of the signal, and the most informative moment in each cycle is not the firing, but the transition, the precise point at which the hyperpolarization lifts and the neurons are released, poised, ready to receive whatever the hippocampus sends in the first milliseconds of the new upstate. In that transition, brief and recurrent as a heartbeat across the length of the night, the day's experiences begin their long migration from the temporary to the permanent. What the body does during all of this is lie still. The muscles are quiet, the eyes are closed. The breath is slow and regular, deeper than it is in REM, less variable than it is in waking. From the outside, nothing is happening. From the inside, at the scale of the oscillation and the ripple and the spindle, the night is full of coordinated, specific, purposeful exchange. Not purposeful in the sense of intention, but purposeful in the sense of precision, in the way a physical system optimized over a very long time tends toward precision, the way water finds the gradient and the crystal finds its form. The medial prefrontal cortex, receiving the night's deliveries, does not know that it is being updated. There is no awareness during slow-wave sleep, no sense of information arriving, no experience of filing or transferring or consolidating. The process runs without supervision, without effort, and without any participation from conscious experience. It runs because the architecture of the brain is built for it, because the slow oscillation, the spindle, and the ripple are part of the brain's structure in the same way that the ventricles are part of the heart structure, built to perform a specific function when the conditions that activate that function are present. And those conditions are present every night in the long dark hours before the later sleep cycles begin to shorten the slow wave periods and lengthen something else. The filing system runs most intensely in the first half of the night, when the delta waves are tallest and the oscillations most regular and the spindles most densely packed into the upstates of each cycle. It is in these hours that the most recently formed and most urgently fragile memories make their first significant moves from the hippocampus toward the cortex. The transfer is not complete after one night. It will continue across the nights that follow, each one adding another layer of cortical strengthening, another reduction in hippocampal dependence, another increment of permanence. But the first night is when the critical early stages of that transfer begin. In the rhythms that the sleeping brain generates for exactly this purpose, cycling through the dark in their nested hierarchy, patient and precise and continuous, while the body rests and the mind drifts somewhere that has no address and leaves no record. The medial prefrontal cortex grows a little more capable. The trace in the cortex is a little stronger, and the experience that a waking brain received as something vivid and immediate, written in pencil, has begun its long, slow process of becoming something carved, the kind of memory that outlasts the original moment by years, that survives the loss of the hippocampus, that returns in the middle of an ordinary afternoon, carrying all of the quiet weight of having been thoroughly, patiently, and silently filed. Deep in the brainstem, smaller than a thumbnail, there is a cluster of cells that fires almost without pause during every waking hour. It fires when light touches the eyes. It fires when a voice rises in the next room. It fires when the body registers cold, or hunger, or the small shock of an unexpected sound. Through all of this, it keeps its rhythm, steady and persistent, sending its signal upward into the forebrain in a continuous chemical stream. And then, at the edge of each REM period, it slows, the firing tapers, the signal thins, and finally, in the span of a few seconds, the cluster falls nearly silent. Not dead, not absent, but quieted in a way it is not quieted at any other time in the entire day. This structure is called the locus coeruleus. It is the brain's primary source of norepinephrine, the chemical that keeps the forebrain primed for alert response. During waking, it bathes the cortex and the amygdala in this signal continuously, setting the tone of vigilance that underlies every emotional reaction, every rapid appraisal of threat, every memory of something that hurt or startled or mattered. When the locus coeruleus fires, norepinephrine rises. When norepinephrine rises, the amygdala amplifies its response to emotionally charged input. The memory of something frightening is stamped harder into the tissue of the brain when this signal is running strong. At the onset of REM sleep, that signal withdraws. The the reduction is not small or gradual over hours. It is steep and specific, falling by as much as 80 or 90% compared to waking levels. The cortex and the amygdala, still metabolically active, still processing and replaying the memories of the day, find themselves doing so inside a brain that has been chemically stripped of the thing that would normally make that processing alarming. The content remains, the alarm is quieted. This is not an accident of sleep architecture. It is the condition that makes REM sleep's most important work possible. The amygdala during REM sleep is not a passive structure. When researchers have placed people in scanning machines that measure blood flow and metabolic activity through the sleeping brain, they find the amygdala running at intensities 20 to 30% higher than it reaches during ordinary waking. It is coupled tightly to the hippocampus, which is replaying the sequences of the day, and to the anterior cingulate cortex, which integrates emotional meaning with the context of experience. This is the same distributed network that first processed the emotional events of the day during waking, the same geography. Reactivated, the amygdala is not resting, it is working, and working hard. But it is working inside the strange neurochemical shelter that the silenced locus coeruleus provides. The adrenergic amplifier is off. The chemical that would turn emotional replay into re-traumatization is almost absent. And so the brain can revisit what happened, can process the emotional content of the memory at high intensity without triggering the same cascade of arousal that the original event provoked. The memory comes back. Its charge is handled differently this time, in a brain equipped with a different chemistry. The result, across a full night, is a kind of recalibration. What arrives at dawn is not a faithful copy of what was encoded the evening before. The factual content of the memory tends to hold, but the emotional weight attached to it, the visceral sharpness of the feeling, the adrenergic edge, is often reduced. The event is still remembered. It is no longer, in quite the same way, relived. This process was demonstrated with precision in a series of human studies. Participants came into a sleep laboratory and learned a set of emotionally charged stimuli, images and associations designed to provoke a measurable fear response. Their amygdala reactivity to each stimulus was assessed before sleep and then again the following morning. The group that slept a full night showed significantly reduced amygdala reactivity to the same material in the morning. The content had been retained. They could still identify and recognize everything they had learned, but the emotional response the material provoked had been genuinely attenuated. Sleep had not erased the memory. It had, in some measurable physiological way, taken the edge off it. Then the researchers selectively deprived a separate group of REM sleep, using EEG monitoring to detect the electrical signature of REM onset and waking the participants each time it appeared, before allowing them to return to the other stages of sleep. Their total sleep time was comparable. Their slow-wave sleep remained largely intact. Only REM was specifically removed, and the following morning, the attenuation was gone. Amygdala reactivity to the fear-conditioned material was unchanged from the evening before. The memory was no less charged than when it was encoded. Without REM, whatever process normally softens the emotional imprint simply had not occurred. The implication sits quietly in those numbers. The brain is not a passive storage device, faithfully preserving experience in the same emotional state in which it was laid down. Every night, while the body lies still and the locus coeruleus rests, the sleeping brain is actively editing the emotional register of the day's events. The editing depends on REM. REM depends on the silence of a small cluster of cells in the brainstem, and that silence is, in a precise physiological sense, the most restful thing the sleeping brain produces. The locus coeruleus does not fall silent for the same stretch of time each night. In the first REM episode, it quiets for perhaps 10 or 15 minutes, a brief window opened early, but the architecture of the night tilts the balance toward REM as the hours pass and the later REM episodes grow longer. By the final episode of the night, the locus coeruleus may rest for close to an hour. The window of emotional processing that opens in the first REM period is narrow. The window that opens in the last is wide, extended, deeply sustained. This is why the final hours of sleep before morning are not redundant. They are the hours in which the most accumulated emotional processing occurs, the hours that are most commonly lost to early waking, to an alarm that interrupts before the final REM episode has completed. When those last hours are cut, the emotional content that would have been processed in the final, longest REM episode stays unprocessed. The record is still there. The adrenergic edge is still attached. The following day carries a residue that a full night would have smoothed. This is not a metaphor. It is a measurable difference in amygdala reactivity to emotional stimuli. The brain that loses its last REM episodes wakes up more reactive than the brain that does not. There is a further dimension to what REM sleep does with memory, one that is not about emotion at all. It is about association, the discovery of connections across experiences that have not yet been consciously linked. While the hippocampus and associated cortex replay the sequences of the day during NREM sleep, REM sleep appears to do something different with the same material. Not deep sequential consolidation, but a wider, less linear integration, a kind of loose coupling between memories that share structure or context, but were encoded at different times in different situations. The evidence for this came from an experiment in mathematics, where participants were given a sequence of operations to perform on strings of numbers, a procedure with hidden structure, a shortcut that, if noticed, would reduce the problem to a single step. The participants were not told the shortcut existed. They simply practiced the procedure, making it automatic. Some then slept before being retested. Others stayed awake for the same interval and returned to the task. The difference was clear. Those who had slept were substantially more likely to have discovered the hidden rule on their own. Something in the interval of sleep had reorganized their representation of the task in a way that revealed its underlying structure. The knowledge they had was the same. The access they had to its deeper pattern had changed. Sleep had not added information. It had reorganized what was already there into a form from which the insight could emerge. The REM duration specifically predicted who made the discovery. The participants with the most REM sleep were the most likely to wake with the insight they had not possessed when they closed their eyes. This was not slow-wave sleep doing the work. It was not simply total sleep time. It was the accumulation of REM episodes, of the specific neurochemical and electrophysiological state that REM provides, that correlated with the moment of understanding. Why REM might perform this kind of integration remains an open area of investigation. One possibility involves the distinctive cholinergic state of REM sleep, a brain saturated with acetylcholine and stripped of norepinephrine and serotonin, a combination that favors associative processing over the more linear, sequential replay that characterizes slow-wave sleep. Another possibility involves the looser, more wide-ranging pattern of reactivation that REM replay appears to produce, less temporally faithful to the original sequence than the compressed ripple-driven replay of NRM, more capable of linking elements from different stored experiences that share a common structure. The honest answer is that both possibilities have evidence, and neither has displaced the other, and the precise mechanism by which sleep turns stored knowledge into accessible insight has not been settled. What has been settled is that it happens. The mathematical shortcut experiment is not alone. A range of related findings converges on the same general picture. Sleep, and specifically REM sleep, does something to the organization of memory that waking does not do in the same way. And one of the things it does is increase the accessibility of connections and patterns that the waking brain had laid down without recognizing. There is a reason the phrase sleeping on a problem has persisted in human language across cultures and centuries. It is not a metaphor. It is an accurate description of a real cognitive process. The problem presented to the brain before sleep is a different problem in the brain that wakes from it. The input has not changed. The encoding has not changed. What has changed is the map of connections in which the problem sits, subtly restructured by REM, made more navigable in the night. The amygdala during REM sleep is one of the most metabolically active structures in the entire sleeping brain. Its high coupling to the hippocampus and the anterior cingulate during REM is not uniform across the night. It intensifies with each successive REM episode. As each episode grows longer, and the locus coeruleus stays silent for a progressively extended stretch. The emotional memories of the day are not all processed equally. The ones with the highest initial amygdala response, the ones that provoked the strongest encoding, are the ones most likely to be reactivated in REM. The brain is not random in what it revisits. The emotionally significant material tends to be prioritized, replayed more frequently, and ultimately across the arc of a full night, tended more thoroughly. This has practical consequences for what waking life feels like the next day. A person whose sleep is structured normally, whose REM episodes complete in their natural progression toward the extended bouts of the final hours, wakes with a different affective tone than a person whose REM has been shortened or interrupted. The emotional events of the preceding day feel, in a measurable sense, less charged. Not forgotten, not irrelevant, but handled, passed through a process that has taken what was raw and made it, at the level of neural response, something calmer. The clinical picture of what happens when this system is chronically disrupted is not a gentle fade. Conditions defined by intrusive emotional memory, the re-experiencing of traumatic events with full adrenergic force, as if the original event were happening again rather than being remembered, show abnormalities in exactly this system. The noradrenergic suppression that healthy REM provides is incomplete in trauma survivors experiencing persistent intrusion. The locus coeruleus fails to fall quiet in the way it should. The amygdala processes the replayed material at full adrenergic intensity rather than in the chemically sheltered state that would allow attenuation to occur. The emotional memory is revisited without being processed. The replay becomes re-traumatization rather than resolution. A drug that reduces norepinephrine signaling at alpha-1 adrenergic receptors, originally developed for blood pressure, was found through careful clinical observation to reduce nightmare frequency and the frequency of intrusive re-experiencing in trauma survivors. The drug is called prazosin. It works by dampening norepinephrine's effect on the very receptors that during disturbed REM sleep remain too active. It extends and deepens the neurochemical quiet that healthy REM sleep provides naturally. The result is that REM replay, with noradrenergic signaling reduced pharmacologically in ways that mirror the natural suppression of the locus coeruleus, begins to function more like the attenuation process it was built to be. The fact that mimicking the chemistry of healthy REM sleep therapeutically produces relief in people whose natural REM processing has been disrupted says something about what that chemistry is for. The locus coeruleus falls silent during REM because emotional memory needs to be processed without adrenergic amplification. The attenuation that follows is not incidental to REM sleep. It is part of what REM sleep is for, as clearly as anything else that can be said about it. The connections between the hippocampus and the amygdala run in both directions. The amygdala modulates what the hippocampus encodes during waking. Emotionally arousing experiences are encoded more deeply, tagged with a signal that marks them for stronger consolidation during sleep. The hippocampus, in turn, replays those tagged sequences back to the amygdala during REM, providing the content for the amygdala's overnight processing. The two structures are in conversation across the entire arc of a day-night cycle. Waking activates the conversation. Sleep completes it, quieting the adrenergic noise that would otherwise prevent completion. This is why REM sleep cannot be cleanly separated from the health of emotional life. A few nights of sufficient REM do not make a resilient emotional system. The effects accumulate, and the debt from disrupted nights also accumulates. The recalibration performed each night is incremental. A single night's work reduces the emotional charge of the day's most salient experiences by some fraction. A full week of nights completes what a single night begins. And conversely, a single night of significant REM disruption leaves a residue that the following night begins to address, but may not entirely resolve. The body keeps this account quietly without drawing attention to it. There is no conscious awareness of the amygdala's work during REM. There is no sense of effort, no experience of the replay, no memory of the processing that occurred. The person lying still with their eyes moving beneath closed lids is undergoing a genuine neurobiological procedure. Emotionally significant content is being reprocessed. Associations are being extended. The adrenergic charge attached to the day's hardest experiences is being reduced, and none of it is available to introspection. It happens entirely below the surface of any conscious experience the sleeper might report in the morning. The morning itself carries the evidence, not in memory, not in any recalled image of what occurred during the night, but in the changed quality of effective response to the preceding day's events. The person who slept fully and well does not remember the emotional processing that happened during the night. They experience only its outcome, the morning's calmer relationship to whatever was difficult the day before. There is something in this that bears reflecting on slowly. The brain does not require awareness to perform its most important regulatory work. The emotional editing that occurs across a full night of healthy REM sleep happens entirely without supervision. The brain revisits what was hard, processes it through a chemistry specifically calibrated for that purpose, attenuates what needs attenuation, integrates what can be integrated, and releases what has been handled. No input from the sleeping person is required, no instruction, no deliberate effort. The work is done in the dark by a system that has been performing this function for longer than there have been words to describe it. The process that extracts a mathematical insight from a night of sleep operates by a similar principle. The waking mind encountered the structure of the problem. It practiced the procedure without perceiving the hidden pattern. Something in the landscape of stored knowledge held the pattern encoded but not accessible. During sleep, and specifically in the accumulated hours of REM sleep, the map of associations was rearranged. Connections that had not been active together were brought into proximity, tested, found to hold. And in the morning, the structure of the knowledge was different. The insight that the waking brain had worked the problem without finding was simply there, available, as if it had always been present and the night had only made the path to it clear. In this sense, REM sleep is doing something to memory that goes beyond storage. It is not simply preserving what was encoded, it is working on the relationship between encoded items, discovering what connects them, what pattern runs through them, what the structure beneath the surface content turns out to be. The mathematical insight is one demonstration of this, but the same process applies at varying degrees to any night's accumulated experience. The day's events do not simply persist unchanged into the following morning. They have been worked on. Their relationships to other stored experiences have been explored and extended. The map is slightly different. The knowledge sits in a slightly different configuration than the one in which it was laid down. The person who wakes in the morning is not the same person who went to sleep, not because of any dramatic transformation, but because the configuration of their stored experience has been subtly rearranged. A few connections are slightly stronger, a few emotional charges are slightly lower, a few patterns that were encoded without being perceived are now slightly more accessible. The changes are small, measured in fractions of a % difference in reaction time or amygdala response, accumulated across nights, across months, across years. They are the substance of how experience slowly becomes wisdom, and how the hardest things that happen slowly become the things that can be lived with, rather than only endured. None of this requires dreaming to be consciously experienced. The dreams that occur during REM sleep, vivid, emotional, often strange in their combination of familiar and unfamiliar elements, may be a side effect of the processing that is underway, or they may serve their own function, or both. The relationship between dreaming as a conscious experience and the neural work occurring during REM sleep is genuinely unresolved. What is clear is that the neural work occurs regardless of whether any dream is remembered in the morning. The attenuation happens, the associative integration happens, the locus coeruleus falls silent, and then rises again. All of this leaves its trace in the brain's changed configuration, whether or not a single image from the night persists into waking. The silence of the locus coeruleus each night is not dramatic. It is not visible or audible or felt. It is simply a small cluster of cells deep in the brainstem that has been firing steadily for all of the waking hours, gradually releasing its grip, slowing, pausing, going still. The cortex that has been bathed in its signal for the entire day gradually feels the signal withdraw. The amygdala, whose emotional intensity depended partly on that signal, finds itself in a different chemical environment, quieter in a specific and consequential way. In that quiet, the day's emotional residue is met again, held differently, and gradually released, not erased. The memory will remain, not ignored. The amygdala is among the most active structures in the sleeping brain, but processed in the specific sense that the adrenergic amplification that originally attached to the experience has been reduced, so that when the memory is retrieved in the morning, it arrives with less of its original force. The event is the same event. The brain that holds it has changed. This is what the locus coeruleus's nightly silence is for. This is what the extended REM episodes of the final hours of sleep are for. The long REM bouts that occupy the last 90 minutes before waking, the ones most vulnerable to being cut short by an early alarm, or by the accumulated fragmentation of a difficult week of sleep, are the bouts during which the most sustained, the most complete, the most thorough emotional processing occurs. The brain saves its longest and most elaborate REM work for last. The hours when the night is nearly over are the hours when the locus coeruleus has been silent for its longest continuous stretch, and the amygdala and hippocampus are most deeply coupled, and the emotional recalibration of the day's hardest material is running at its fullest. The researchers who demonstrated the relationship between insight and REM duration found that the mathematical shortcut was most likely to be available in the morning after the nights with the most accumulated REM sleep. Not the nights with the most slow-wave sleep, not the nights with the most total sleep time, but specifically the nights with the most REM. The relationship was specific, and it pointed towards something that the plain observation of sleep had not, on its own, been enough to establish, that the accumulation of REM episodes across the night is not redundant repetition of the same process. Each successive REM episode is longer than the last, and the work each performs on the landscape of memory extends what the previous one began. The final episode does not replicate the first. It completes something the first only started. Somewhere in the last quiet hour of the night, in the long sustained REM episode that precedes waking, the locus coeruleus rests at its deepest. The cortex runs at near waking intensity. The hippocampus and amygdala move through material that has been partially processed through the earlier, shorter REM periods and now receives the longest, most sustained window of noradrenergetically quiet attention the night can offer. The mathematical structure that was hidden in the procedure becomes visible. The fear that attached to the difficult image loses one more fraction of its adrenergic edge. The connection between two stored experiences, laid down in different weeks, in different contexts, linked by a common thread that no waking process had fully drawn, is gently strengthened. And then the locus coeruleus begins to fire again, slowly at first, then with its full waking rhythm restored. Norepinephrine rises back through the forebrain. The cortex brightens toward the particular tone of waking alertness. The body begins to stir. The night is ending, and the brain that opens its eyes has done something the brain that closed them could not do. It has taken the raw material of the previous day's experience and worked it, quietly in the dark, into something slightly more resolved. The mathematical problem that seemed opaque the evening before yields its hidden rule without effort in the first waking hour. The difficult conversation from the previous afternoon sits in memory with slightly less charge than it carried going into sleep. The connection that had been present in the stored knowledge but not yet accessible announces itself, simply and without ceremony, in the mind of the person making coffee in the morning light. None of it announces its origin. The insight does not arrive labeled as a product of the night's REM processing. The emotional softening does not feel like the result of a neurobiological procedure performed in the brainstem. They simply feel like the morning, and like being rested, and like the ordinary experience of a night having passed. The brain completed its work without signatures. It rarely asks for acknowledgement. The evidence it leaves is only the changed quality of how the world is met when light returns. The locus coeruleus, deep in the brainstem, fires on. The day resumes, somewhere in the night just past, in the long-sustained quiet of the final REM episode. The day before it was gently and irreversibly changed. In a laboratory in Wisconsin, in a room that smells of cold electronics and filtered air, a researcher sits in front of a monitor at 2 a.m. and watches an image no one in the world had ever seen before. On the screen, rendered in false color, are the cross-sections of mouse cortex prepared by serial electron microscopy. Tissue sliced thinner than a wavelength of visible light, each slice imaged, the images stacked into a three-dimensional reconstruction of something no larger than a grain of sand. Inside that reconstruction are synapses, thousands of them, each one measured, each one assigned a number representing the area of contact between two neurons, the physical footprint of a connection, precise to the nanometer, smaller by several orders of magnitude than anything a light microscope could resolve. And those numbers, when compared between animals that had slept and animals that had not, told a story that the field had been circling for 2 decades without being able to see directly. The synapses were smaller after sleep. Not all of them, and not uniformly, but on average across nearly 7,000 measured contacts, the axon-spine interface area was about 18% reduced in the animals that had slept compared to those that had stayed awake. 18%, not a dramatic collapse, a quiet, systematic reduction, the kind of change that would be invisible to any instrument less precise than the one in that room, and that would leave no visible mark on the animal, no behavioral sign, no signal on a surface recording, just the physical architecture of the brain's connections, silently reshaped across the hours of darkness. This is what sleep does to the structure it has been building all day. It adjusts the building, carefully, at the smallest scale at which the building exists. The hypothesis that predicted this finding had a name. It was called synaptic homeostasis, and it had been proposed by Giulio Tononi and Chiara Cirelli in 2003, not as a settled fact, but as a framework, a way of thinking about why slow-wave sleep might be the specific stage of the night most tightly linked to memory performance. The core idea was simple in outline, though its implications reached into nearly every corner of neuroscience. While an animal or a person is awake, experience drives synaptic strengthening. Every potent encounter, every learned association, every moment that leaves a trace, leaves that trace partly through the physical enlargement and sensitization of synapses, the molecular events that had been inscribed during the day across billions of contact points throughout the cortex. By the time the day is over, the network has grown more potent in 1,000 places, more sensitive, more expensive to run. The expense is real. Synapses require energy to maintain. More precisely, they require a constant supply of the molecules that keep the postsynaptic machinery assembled, the receptor proteins anchored in their slots, the scaffolding proteins holding them in place, the cytoskeletal elements maintaining the shape of the dendritic spine itself. A strong synapse is a metabolically demanding synapse, and a brain that added strength at synapses throughout every waking hour without ever pulling back would eventually reach a state in which the signal-to-noise ratio of its own activity collapsed, where everything was strong and therefore nothing stood out, where learning had been preserved, but new learning had become nearly impossible because the machinery was already saturated. Slow-wave sleep in this picture is the global correction, the slow oscillations that move through the cortex during deep sleep. Those great cycling waves of firing and silence are the physical instrument by which the brain steps back from the potentiation of the day and returns synaptic weights toward a sustainable baseline, not erasing what was learned, renormalizing it, preserving the relative pattern of strengthening, which synapses are stronger relative to which others, while reducing the absolute values across the board, so that the next day begins with room to write again. The electron microscope images in Wisconsin were the first direct structural confirmation that this renormalization is not merely a functional metaphor, but a physical event. Synapses actually get smaller. The protein scaffolding actually contracts. The axon-spine interface actually shrinks. The sleeping brain is not idle. It is performing a precise mechanical operation on its own hardware at the scale of individual molecular junctions across the entire cortex, while the animal lies still and the room outside is dark and quiet. What remains after the operation is a brain whose connections are slightly smaller on average, whose energy costs are somewhat reduced, and whose capacity for new potentiation has been partly restored. The memories of the day survive this process. The pattern of which synapses are relatively stronger persists. The relative landscape of the learned trace remains, even as the absolute terrain is lowered. Sleep does not erase the day. It files it more efficiently, compresses it, makes room for what comes next, the way a careful archivist does not discard what has been recorded, but rearranges the storage so the shelves can hold more. The slope of the slow-wave EEG signal at the beginning of the night tracks this. On the first night, after a day of dense learning, the slow waves at sleep onset are steeper and taller, high amplitude, high synchrony, reflecting the degree of synaptic potentiation that has accumulated and is now being worked through. On a night after a day of low cognitive demand, the waves are lower, shallower, quicker. The brain calibrates the depth of its own maintenance according to how much maintenance is needed. It reads the load and adjusts the depth of the cleaning accordingly, in a self-regulating process that requires no oversight and no awareness, running entirely below the level of any experience the sleeper will ever have. And then, while the synapses are being renormalized, something else is happening, something that operates at a completely different physical scale, using a completely different mechanism, performing a completely different kind of maintenance. Something that was not discovered until 2012, and that changed the way the sleeping brain was understood to work. There is a system that runs through the brain like a hidden river. It has no dedicated anatomy visible to the naked eye, no distinct channels lined with specialized cells, no valves, no obvious structure. What it has instead are gaps, the narrow spaces between cells, the channels that run alongside blood vessels as they thread through brain tissue, the mesh of the extracellular matrix through which fluid can percolate under the right conditions. In those conditions, the conditions under which the fluid actually moves through the brain in volume, doing real work, are the conditions of sleep. The system was described first in mice, using a method that required the brain to be alive and intact. Two-photon microscopy, in which a laser excites fluorescent molecules injected into the cerebrospinal fluid, making the fluid itself visible as it moves through tissue in real time. What the images showed when they were first analyzed was a flow, cerebrospinal fluid entering the brain, not through the ventricles as the old picture had it, but along the spaces surrounding the arteries, narrow channels wrapped in a layer of astrocyte membrane, the perivascular space, through which fluid was being driven inward as the arteries pulsed, flowing through the tissue, and then exiting along the spaces surrounding the veins, draining outward and downward, ultimately toward the lymphatic vessels in the neck, a river with a very slow current moving through an extremely narrow landscape, driven not by a pump, but by the rhythmic pressure of a beating heart transmitted through vessel walls. The name given to this system carried a quiet acknowledgement of the cells that make it possible. The glial cells, the astrocytes, whose membrane extensions wrap the blood vessels and whose water channels line the perivascular space, determining how easily fluid crosses from the vascular wall into the tissue. The glymphatic system, glial plus lymphatic, a word that names the partnership between two kinds of tissue that no one had previously thought to look for, working together in this way. The astrocyte's role is structural, but precise. At the point where the perivascular space meets the brain, parenchyma, the tissue proper, where neurons and glia live, the astrocyte end feet create a boundary, dense projections of astrocyte membrane pressed flat against the outside of the blood vessel, forming a sleeve. And in that sleeve, the membrane is packed with water channels, proteins called aquaporin 4, present at higher density in the perivascular end feet than almost anywhere else in the brain, each one a narrow pore through which water molecules, and carried with them dissolved solutes, can cross from the perivascular space into the interstitial fluid of the brain. A single astrocyte end foot, seen under the electron microscope, looks like a dense, slightly flattened pad of membrane, closely applied to the surface of a vessel that is itself barely wider than a red blood cell. The aquaporin 4 proteins are too small to see directly in most preparations, but their presence is inferred from the speed of water movement, and their function is demonstrated most clearly by the experiments in which they are absent. In mice lacking aquaporin 4, glymphatic transport slows dramatically. The river still exists, but the crossing from the vessel wall to the tissue is obstructed, and the solutes that would have moved through accumulate instead. Those solutes are the metabolic byproducts of a day of neural activity, proteins produced and shed by neurons in the ordinary course of their function. Small peptides, fragments of larger molecules that have been processed and released into the interstitial space, and among them, in a brain that is aging or has been stressed or has simply been running for many hours, the peptides that aggregate into the structures associated with neurodegeneration, amyloid beta, and the protein known as tau. Amyloid beta is produced continuously by neurons as a byproduct of normal cellular metabolism. It is not a pathological molecule in itself. It is simply present always in the interstitial fluid of any living brain. The question that matters is not whether it is there, but whether it is cleared before it accumulates to concentrations at which it begins to aggregate into the clumps that are damaging to the tissue around them. And the answer to that question depends in large part on how well the glymphatic system is running. The rate of glymphatic flow through the brain is not constant. It rises and falls across the day, and the relationship between sleep and glymphatic activity is not subtle. In the sleeping mouse, the interstitial space, the physical gaps between cells through which the fluid must percolate, expands to roughly 60% greater volume than in the waking brain. This is not a passive collapse when the brain's activity drops. It is an active structural change driven by the redistribution of water in astrocytes across the transition from waking to sleep, as those cells shift water from their interior into the extracellular space, widening the channels through which glymphatic fluid must pass. The resistance drops. The flow increases, the river runs faster. The consequence is a doubling, approximately, of convective flow rate through the sleeping brain's tissue, fluid moving through wider channels, carrying dissolved material with it at twice the rate it moved during waking hours. Amyloid beta concentration in the interstitial fluid, which had been building since the animal woke, begins to fall. The peptide moves with the fluid into the perivenous drainage, out of the brain toward the cervical lymphatics, eventually into the body's broader circulation, where it is processed and cleared by the liver and the kidneys. The brain's own chemistry is being diluted, refreshed, cleaned by the simple physical movement of fluid through its substance. This process has now been observed in humans through methods that can track amyloid burden across a night rather than a moment. In one study that used dynamic imaging to measure amyloid levels before and after sleep, a single night of total sleep deprivation produced a measurable increase in amyloid beta in two specific regions, the hippocampus and the thalamus. The hippocampus, the structure at the center of memory consolidation; the thalamus, the relay that gates sensory information and generates the sleep spindles through which memories are transferred to stable cortical storage. These two regions accumulated more amyloid in one sleepless night than they would have during a normal night of sleep. Not a catastrophic accumulation, not visible damage, but a measurable shift in living human tissue in the direction that decades of work have linked to neural vulnerability. One night, one mist clearing cycle, a detectable change in the chemistry of the structures the brain depends on for the functions that matter most. The glymphatic system does not have the kind of elegant, nested timing of the spindle ripple slow oscillation hierarchy. It does not fire in bursts, does not replay memories, does not transfer specific content from one region to another. It operates instead as a slower, more diffuse process, a chemical tide, rising through the hours of sleep and receding with waking, leaving the tissue slightly cleaner than it found it, making the conditions more favorable for the next day's work. It is maintenance in the most literal sense, the washing of the floor while the building is closed, so that the instruments on the benches can function without the interference of accumulated debris. The two processes, synaptic downscaling and glymphatic clearance, do not operate in sequence, one after the other. They unfold simultaneously across the same hours, within the same tissue, their timescales overlapping, while the slow oscillations cycle through the cortex, organizing the renormalization of synaptic weights and driving the transfer of memory traces towards stable cortical storage. The astrocytes are widening the interstitial channels, and the cerebrospinal fluid is moving through, carrying away the molecular residue of the day's activity. The brain during deep sleep is performing two kinds of housekeeping at once, structural and chemical, one in the synaptic architecture and one in the fluid that bathes it, each serving the other in ways that are only beginning to be understood. The synaptic downsizing may itself contribute to glymphatic efficiency by slightly reducing the volume of dendritic spines and clearing some of the physical obstruction from the interstitial channels. The glymphatic flow may support synaptic maintenance by removing the accumulation of metabolites that would otherwise interfere with the molecular machinery doing the renormalization. The two systems share the slow wave stage of sleep as their primary window of operation, because slow wave sleep is when the cortex is most deeply synchronized, when the body is most still, when arterial pulsation is most regular, and when the astrocytes have made the architectural adjustment that allows the fluid to move. Slow wave sleep is not merely when memories are consolidated. It is when the physical substrate of memory is serviced. And this matters because the substrate is not permanent. It is biological. It is made of proteins with finite lifespans, maintained by cells that require energy, operating within a chemical environment that must be actively regulated to remain compatible with their function. The neurons of the hippocampus and cortex are not inert storage media. They are living cells, dependent on the integrity of their membranes, the availability of their receptor proteins, the stability of their cytoskeletal scaffolding, the continuous clearing of the metabolic waste their own activity produces. Memory is not written on something that lasts forever. It is written on something that must be maintained, and the maintenance happens at night, in the dark, while the mind that holds the memories is not attending to them. There is a quality of great patience in this picture, not the patience of waiting. Nothing here is waiting, but the patience of a process that runs without oversight, without awareness, without any attention paid to it from any level of mind. The astrocytes do not know they are clearing amyloid. The cortical neurons do not experience the reduction in synaptic weight as a loss. The cerebrospinal fluid does not know it is doing work. And yet the work is done across every night of every life in the silent architecture of a body that has found ways to care for itself that are more precise, more thoroughgoing, and more elegant than anything that could be deliberately arranged. Consider what the night looks like from the outside of the skull. Nothing. A person lying still, breathing slowly, eyes closed, showing every sign of inactivity. And inside, the slow oscillations cycling at roughly 1 per second across the surface of the cortex, each one an up state followed by a down state, the neurons firing in concert and then falling silent in concert, the rhythm moving across the cortical surface like a wave across still water. Nested inside the upstates, sleep spindles, brief self-sustaining bursts of oscillation generated in the thalamus and broadcast across the cortex, each one lasting a second or two, each one a window of opportunity. And nested inside the spindles running in the hippocampus, sharp wave ripples, compressed replays of the day's experience, firing in bursts that last only 100 milliseconds, the day's neural sequences playing through again and fast forward, sending their signal to the cortex where the slow oscillation is already holding the door open. And simultaneously, in the gaps between all of these neurons, fluid is moving, slowly, steadily, driven by the pulse of the blood vessels and facilitated by the water channels in the astrocyte end feet, carrying dissolved amyloid beta toward the drainage pathways, refreshing the interstitial chemistry of the tissue in which all of this electrical activity is occurring. And at the synapses, those small, precise junctions between the neurons, whose firing organizes all of this, a molecular adjustment is underway. Receptor proteins being drawn back from the membrane at connections whose weights have exceeded the local optimum, scaffolding proteins redistributing, the physical footprint of the connection, that axon-spine interface, settling to a slightly smaller value, precisely calibrated to preserve the relative pattern of the day's learning while restoring the margin needed for tomorrow's. 3 processes, 3 timescales, 3 physical mechanisms, unfolding simultaneously in the same tissue, while the person lying on the bed shows nothing at all. The finding that perhaps brings this into sharpest focus is also the one that makes the stakes most immediate. In that study of one sleepless night, healthy adults kept awake for 1 night under controlled conditions. Their brains imaged before and after. The amyloid accumulation was not uniform across the brain. It was concentrated in the hippocampus and the thalamus. These were not random targets. They were the structures most directly involved in the processes that had been running all day. Memory encoding in the hippocampus, sensory gating, and sleep spindle generation in the thalamus. And they were, by that account, the structures that had been working hardest, producing the most metabolic byproduct and most dependent on the nightly clearing that had been withheld. The burden did not reach pathological levels in 1 night. It was not a disease state, but it was a signal, a demonstration in living tissue that the glymphatic system's contribution to brain health is not a background condition that quietly maintains itself regardless of how much sleep a person gets. It is a function that runs when sleep runs and stops when sleep stops, even briefly, even for 1 night. What this means for the long accumulation of a life, for the chemistry of a brain that has been chronically short on sleep for years or decades, is a question the research is still building toward. The answer will not be simple, because the brain is not simple, and its relationship to its own clearance systems is not simple. There are many variables. There are compensatory mechanisms. There are differences between individuals that make predictions difficult. But the direction of the evidence is consistent, and it points toward a relationship between the quality and quantity of sleep and the long-term chemical environment of the brain that is more intimate and more consequential than most people living in a world that undervalues sleep have been given reason to believe. The picture that has emerged from a century of sleep research, from the early EEG recordings that first made the architecture of the night visible, through the electrophysiology that revealed what sleep does to the synapses, through the imaging that first showed the hippocampus replaying the day, through the electron microscopy that measured the synapses before and after, through the two-photon imaging that revealed the glymphatic river is not a picture of rest, it is a picture of work. Deep, precise, coordinated, multi-scale work done by a system of staggering complexity, running without supervision, completing itself in the dark. Rest is not what sleep is. Rest is something sleep makes possible, the feeling of waking with a brain that has been serviced, whose synaptic weights have been adjusted, whose chemical environment has been refreshed, whose memory traces have been transferred from temporary to more stable storage, whose emotional charges have been partly attenuated. The lightness of a good morning is not the absence of something. It is the presence of something, the evidence, felt in the quality of thought and mood and readiness, that the maintenance was carried out and the instrument has been returned to the condition in which it can work. The night is long for a reason. The first cycles, heavy with slow-wave sleep, do the deepest structural work, the most powerful slow oscillations, the most active spindle bursts, the greatest glymphatic flow rate, the steepest slope of synaptic renormalization. The later cycles, lengthening into the longer REM episodes of the early morning, do a different kind of work, integrating what has been transferred, reprocessing the emotional weight of the day's experiences, drawing connections between what was learned today and what was known before, building the broad associative structures that support insight and flexible thinking. Each part of the night is doing something the other parts cannot. And cutting any part short, losing the early deep sleep, losing the late REM, means losing a specific kind of maintenance that nothing during the waking hours can replicate or replace. The sleep spindle that forms in the thalamus and spreads across the cortex during a normal night is not something the waking brain can generate. The slow oscillation that coordinates hippocampal replay with cortical consolidation exists only in the context of sleep, specifically slow-wave sleep, and the electrochemical conditions that make it possible are incompatible with waking. The glymphatic flow that clears the interstitial space depends on the architectural change in astrocyte volume that sleep initiates and waking reverses. These are not processes that run at reduced capacity when sleep is shortened. They are processes that require sleep as their precondition, and in the absence of that precondition, they do not run. This is the plain fact at the center of everything that has been described across this long, quiet night. Memory is not a product of the day. It is a product of the day and the night that follows. The experience encodes something fragile in the hippocampus, a trace that is not yet stable, not yet integrated, not yet filed where it will survive. And then sleep takes that trace and works it, replays it in the compressed language of ripples, transfers it through the nested timing of spindles and slow oscillations to the cortex, where it grows slowly more stable, more independent of the hippocampus that first held it, adjusts the synaptic weights of the connections that carry it, so that the signal can be found again without the noise of an oversaturated system obscuring it, and all the while, the chemistry of the tissue through which those signals move is being maintained, the amyloid cleared, the interstitial space refreshed, the molecular environment of the neurons kept within the range in which they can continue to do this work across the years and decades that lie ahead. The trace left by a Tuesday afternoon, a conversation, a view out a window, the specific quality of light on a surface, the name of a person newly introduced, will not survive by virtue of the intensity with which it was experienced, or the care with which it was attended to, or the deliberateness of the encoding. It will survive if it survives, because the night that followed gave it what it needed: the replay, the transfer, the consolidation, the maintenance of the substrate in which it was written. It will survive because sleep took it and filed it. This is a process that has been running in sleeping animals for as long as there have been animals that sleep, which is by current evidence as long as there have been animals at all. It predates the kind of experience that could have designed it or intended it or known it was happening. It runs in a mouse, in a bird, in a fish, in an octopus, in forms different in their details but convergent in their function, across hundreds of millions of years of independent evolution, because a brain that stores information and then maintains and consolidates that storage during a period of reduced sensory input is a brain that functions better than one that does not. The selection pressure does not know it is building a filing system, but across enough time, what it builds is exactly that. What remains at the end of this long accounting is not something complicated. It is something very simple. The brain, across the hours of sleep, is taking care of the mind, adjusting its architecture, clearing its chemistry, organizing its records. And it does this in the dark without being asked, without awareness, without the mind that benefits from it having any sense that the work is being done, because the mind at that hour is not available to witness it. The cerebrospinal fluid moves slowly through the interstitial spaces of a sleeping brain, carrying with it the dissolved residue of the day, the small molecules, the shed peptides, the amyloid beta that has been accumulating since morning, and it moves toward the drainage channels at the margins of the tissue and out slowly toward the cervical lymphatics, toward the body's wider circulatory system, toward eventual clearance in organs that were built for exactly this purpose. The flow is not fast. It does not rush. It moves at the pace of something that has all night, because it does have all night, and all night is what it needs. The astrocyte end foot, pressed flat against the vessel wall, holds its aquaporin channels open. The fluid passes through. The interstitial space, widened by the architectural change that sleep alone produces, offers less resistance than it did an hour ago when the brain was awake. The amyloid beta concentration in the fluid bathing the hippocampal neurons is slightly lower now than it was at the beginning of the night, and by morning, when the sleeping body stirs and the slow oscillations give way to the lighter, faster rhythms of near waking, the fluid will have done its work, and the channels will narrow again, and the river will slow, and the brain will rise into waking, carrying a slightly fresher, slightly cleaner, slightly better tuned version of the substrate it brought into the night. The memories of the day before will be there, more stable than they were, less dependent on the hippocampus, distributed more broadly across the cortex, available for retrieval in a way they were not 12 hours earlier.